As he said, I am finishing my second year as a PhD student in Yachi Ho's lab at Yale. So today I will start by describing the use of single-cell technologies in viral infections, both the promising and challenging aspects that they present. And then I will explain the current major challenge, the rarity of infected cells, after giving some background about HIV infection. I will demonstrate how transcriptome analysis of HIV-infected cells has allowed researchers to identify mechanisms of HIV persistence and highlight the importance of understanding immune cells in the lymphoid system for immune control mechanisms. And then finally, I will focus on a recently published paper from Samuel Kaiser in Alex Shalek's lab that uses single-cell RNA sequencing analysis to reveal key immune pathways in acute HIV infection. And then finally, I'll provide some perspectives about future studies. So first, the study of viral infections has and will continue to benefit immensely from the application of high-throughput single-cell technologies. Now, the majority of current single-cell studies on viral infections focus on how viral infections impair the immune system. Typically, viral infections cause systemic immune activation or immune dysfunction. Single-cell technologies provide the resolution to understand these heterogeneous immune responses. For example, single-cell technologies can capture the activation of a wide range of immune responses from innate immunity to adaptive immunity. Second, single-cell technologies can capture thousands of cells to resolve the high heterogeneity of immune responses. For example, even when we look only at one T-cell subset, such as CD4 positive T-cells, they can be activated or resting, exhausted or functional, and different polarization states, such as Th1 or T regulatory, and possess different memory phenotypes, such as naive, central memory, and effector memory. We clearly see single-cell studies are well suited to capture the diverse range of immune responses and the heterogeneity of cellular subsets during infection. So the most prominent challenge for single-cell analysis of viral infection is to characterize infected cells, such as in HIV or in SARS coronavirus 2. The difficulty is that these infected cells are rare. If we apply single-cell technology to cancer tissues or to study embryonic development, every cell captured has a biological meaning. In contrast, in viral infections, such as HIV, only a very small number of cells captured and analyzed in the data are actually HIV-infected cells. We can feel like we're searching for a needle in a haystack. For example, only an average of 1 in a million CD4 T cells in the blood harbor infectious HIV, but sequencing 1 million single cells to capture one cell is far beyond the ability of a typical research budget. Second, identifying these infected cells and distinguishing them from uninfected requires mapping and enriching viral RNA in a dataset, which typically requires in-house assay development. The ability to maximize the efficiency of single-cell technologies in viral infections depends firstly on the selection of good clinical samples enriched with infected cells, and secondly, on the ability to identify the infected cells with a relatively shallow read depth. In spite of this challenge, single-cell approaches are still necessary to fully understand viral infections. In this presentation, I will first use HIV as an example to demonstrate how hard it is to capture HIV-infected cells and provide our thoughts about how to find the most informative clinical samples. I will then discuss a recently published single-cell paper describing immune responses during acute HIV infection. So in untreated HIV infection, HIV infects and destroys a key part of the immune system, CD4 positive T cells, thereby wiping out adaptive immunity. Untreated HIV-infected individuals will develop acquired immunodeficiency syndrome, or AIDS, and death. Antiretroviral therapy prevents new rounds of infection and lowers the levels of viral RNA in the blood to undetectable levels. However, whenever a person stops taking antiretroviral therapy, or ART, the virus will come back and AIDS resumes. This is because HIV inserts itself into the host genome, shown in the integration at the bottom of this figure. We refer to the integrated HIV DNA as the provirus. ART cannot remove this integrated HIV. Most actively infected cells will die of the viral cytopathic effect or immune clearance. However, some HIV-infected cells enter a state of latency when the memory CD4 positive T cells go into their resting memory state. No detectable RNA or protein is produced, so the cell escapes detection from cytolytic immune cells. These latently infected CD4 T cells remain in the body of infected individuals and form a stable reservoir. This reservoir decays extremely slowly, with a half-life of almost 4 years, so complete eradication by decay would take nearly an entire lifetime. As a result, infected individuals must continue ART for the remainder of their lives to prevent rebound and disease development. With 37 million people currently infected with HIV and all requiring ART, the adverse effects of ART drugs and the economic burden of lifelong treatment call the research community to work for a cure. As the latent reservoir is the major barrier to cure, it is critical to understand the reservoir in order to develop curative strategies. We still do not fully understand the mechanisms of latency and reactivation or the maintenance of reservoir cells. The first challenge in understanding HIV-infected cells is that even in all HIV-infected cells, only 10% of them contain infectious HIV, and about the other 90% contain defective HIV. The majority, 90 to 99% of proviruses detected in infected individuals are defective. Some proviruses are hypermutated as a result of cellular factors, while others contain mutations in necessary regions of the genome. Many have large internal deletions. Multiple studies have estimated the size of the replication-competent inducible reservoir on the order of 10 to the 8 cells, which could be spread throughout the body. However, these are estimates based on imperfect tests that measure copies of viral genes or look for viral outgrowth to quantify the reservoir. For example, some defective proviruses can still produce viral RNAs, so assuming any cells containing HIV RNA are able to produce infectious virus is not correct. Many assays have been developed to measure the reservoir, but we do not yet have an assay to measure all of the replication-competent inducible proviruses in a sample without including defective proviruses or missing some of the replication-competent proviruses. That leads us to the rarity of infected cells. To identify the most informative clinical samples to study HIV persistence, we first need to understand where to find HIV-infected cells. So far, most studies about HIV persistence have involved the use of blood samples, mainly because of the easy accessibility of obtaining blood compared to obtaining tissues from humans. With the last gift cohort, in which HIV-infected individuals donate their body for research and prompt processing, tissue samples from all human organs were examined to understand where HIV hides. Using HIV DNA as a surrogate for the reservoir, the highest levels are found in the peripheral blood mononuclear cells, or PBMCs, in the gut, and in the lymph nodes. Even in the highest sample, indicated here, there is HIV DNA in about 6 per 10,000 cells, demonstrating this rarity problem. This is even more clear when looking at individuals one by one. Though there is heterogeneity, the primary sites of HIV DNA are the blood, the gut, and the lymphoid tissue. On average, there are about 100 per million cells, or 1 per 10,000 in the peripheral blood, 200 per million in the gut, and 100 per million in the lymph node that contain HIV DNA. These numbers tell us how hard it is to capture HIV-infected cells using single-cell technologies. However, given that more than 90% of HIV DNA captured is from defective proviruses, while only 10% of these cells actually harbor infectious HIV, it is possible that even the tissue samples which have high HIV DNA levels do not actually contain any cells expressing HIV RNA. While autopsy samples allow studying all tissues, they're obviously not easy to get. Another way to study HIV persistence in tissues is to use lymph node and gut biopsies. The samples from human lymph node and gut demonstrated stability after ART treatment in terms of DNA-positive cells. Assuming about 10 to the 9 cells per gram of tissue, these samples contain about 1 HIV DNA positive cell per 10,000, still on the same order of magnitude as the previous study. In comparison to PBMCs, the gut-associated lymphoid tissue, or GALT, contains more HIV DNA and CD4 positive T cells, roughly around 5 per 1,000 cells in this sample, with 8 per 1,000 in the optimal sample, or about 1 or 2 in the PBMCs contain HIV. This suggests we may have a better chance of capturing HIV-infected cells in the GALT than in the peripheral blood, and in samples like these, where ART was not started immediately. Again, since more than 90% of HIV DNA comes from defective proviruses, we still don't know how many cells from these tissues harbor infectious HIV, or even how many can express HIV RNA. Human tissue samples are challenging, so how about animal models? The animal model which can most closely recapitulate HIV infection is an SIV-infected macaque model. In a non-human primate model with simian immunodeficiency virus, similar to HIV, in rhesus macaques, we noticed that this is enriched in lymph nodes and gut during acute infection, and that after treatment, the gut becomes the primary site of the reservoir under ART. HIV DNA-based measurement again does not tell us whether there are any RNA-positive cells that can be captured in these tissues. This problem has been approached by in situ RNA hybridization using RNAscope. The RNAscope amplifies the RNA signal with branching technologies and enhances specificity by amplifying the signal only when 2 adjacent probes are both bound to the target sequence. Using SIV samples as a model, Jake Estey's group used RNAscope and demonstrated high specificity in detecting SIV RNA positive cells with minimal background and false positives in uninfected tissue on the left and infected tissue treated with RNase on the right. This technique is able to detect both SIV RNA inside cells and inside virions bound to the surface of cells, as confirmed by correlation with the quantitative qRT-PCR. The Estes group next applied DNAscope to detect SIV DNA proviruses. DNAscope is much more difficult than RNAscope because one cell may have thousands of copies of SIV RNA, but will only have a single copy of the SIV DNA. In cell lines that were known to contain 1 copy of the provirus, SIV DNAscope results agreed with the expected frequency based on the typical diameter of a cell and the thickness of the tissue section. And DNAscope for HIV works in cell line dilution studies as well. Finally, the Estes group combined both SIV DNAscope and RNAscope to visualize both viral nucleic acids in the same tissue sections using probes specific for DNA and for RNA. Combining DNA and RNAscope shows us which cells have actively transcribed versus latent or defective SIV, though it does not distinguish between those two. By staining adjacent sections, we can compare DNA, RNA, and protein to see which cells are latently infected and actively infected. All 3 measures correlated well in this study. As expected, the number of SIV DNA positive cells is higher than the number of cells expressing SIV RNA or protein, suggesting that either the SIV infected cells harbor a latent or a defective provirus. Beyond calculating the frequency of infected cells, in situ hybridization combined with immunofluorescent staining allows preliminary identification of the immune phenotype of SIV-infected cells. To learn about how infection affects various cell types, we can multiplex DNAscope with immunofluorescence for cellular surface markers. With CD3 to identify T cells, we see infection primarily in these known targets and are able to identify cells containing multiple proviruses, called superinfection, as indicated by the white arrows here. One important benefit of this method is the ability to closely inspect infected cells. The right panel shows a macrophage that has engulfed an infected T cell rather than a directly infected macrophage. All of the tests were performed with tissues from acute untreated infection, samples which will have the highest levels of virus and actively infected cells. To understand the impact of ART treatment on the change in SIV tissue reservoirs, SIV DNA-RNA scope found that RNA-positive cells substantially decreased after ART, suggesting productive infection has indeed been blocked by ART. However, DNA-positive cells decay less drastically, as cells with defective or latent SIV can still persist despite ART. ART-treated samples present a much larger challenge for visualization, since active infection is greatly decreased. When applied to the B-cell follicle of the lymph node in SIV infection, which typically contains many SIV-infected cells during acute infection on the top, DNA-positive cells are nearly undetectable with DNA scope, even though they are detected by PCR. Thus, further improvement of this technology may be needed to successfully analyze ART-treated tissues. Now that we have a technique to visualize the reservoir in tissues, another main question in the field is how are reservoir cells maintained? Do they have long lives? Do they proliferate frequently? Or is it both? Comparing the expression profiles of HIV-infected versus uninfected cells may give a hint about how HIV-infected cells survive. For example, OX40 is found to be enriched in HIV-infected cells. By sorting OX40 positive cells, Matthias Lichterfeld's group found more HIV DNA copies in OX40 positive than OX40 negative cells. These results suggested that OX40 BRCA5 axis could be helping to prevent infected cells from undergoing apoptosis. By using expression of both HIV envelope and GAG as a surrogate for HIV infection, Michelle Nussenzweig's group activated CD4 positive T cells for 30 hours and sorted these activated cells expressing both envelope and GAG. They found a transcriptional signature which suggests cellular survival and suppression of antiviral immunity in infected cells compared to uninfected controls. Using HIV RNA expression as a surrogate for HIV infection, our lab developed HIV sort-seq. In this method, RNA fluorescent in situ hybridization probes detect HIV RNA positive CD4 T cells after 16 hours of activation. Among the differentially expressed genes in sort-seq positive and sort-seq negative cells, genes related to T cell activation and proliferation were upregulated. Therefore, there are many varied mechanisms that could contribute to the maintenance of infected cells. However, it is important to keep in mind that these cells were all reactivated and no longer latent. Thus, it is still not known what truly latent cells look like, since previous detection methods have all required latency reversing strategies. This is one of the main issues to solve in the field. In addition to understanding how infected cells maintain themselves, it is also important to know how infected cells are affected by the immune system. Though many immune cell subsets are involved in viral control, as shown in the figure on the right, the major immune effector which can kill HIV-infected cells is CD8 T cells, highlighted in red. The graph on the right demonstrates that long-term non-progressors, who are individuals who are able to control HIV infection without ART, have a higher frequency of active cytolytic CD8 T cells than individuals who require ART. However, CD8 T cells cannot eliminate all HIV-infected cells. First, chronic HIV infection leads to immune exhaustion, a state in which immune cells progressively lose function as they are exposed to antigen repeatedly over time. The number of HIV-specific CD8 T cells does not correlate with viral load, but with the mean fluorescence intensity of PD1, a marker of exhaustion, correlates well with viral load. Thus, the infection directly affects exhaustion. Second, HIV develops escape mutations to avoid CD8 T cell clearance, illustrated here as filled circles in the diagram of the genome. Third, CD8 positive T cells cannot easily enter particular areas of the body called immune sanctuaries. For example, the B cell follicles of the lymph node cannot be accessed easily by CD8 T cells, unlike the T cell zone, because they lack the homing marker required to enter the B cell follicle. Therefore, in the B cell follicles marked in white by CD20 staining, SIV-infected cells persist and evade CD8 T cell clearance. In this acutely infected SIV macaque model, replication is occurring in the follicles, but is much lower in the T cell zones. Fourth, HIV infection in the gut can lead to destruction of the gut-associated lymphoid tissues, shown on the right, causing permanent injury to the lymphoid structure and irreversible fibrosis. Such structural damage cannot be restored after ART. Lymphocytes lose a niche for homeostasis, proliferation, and replenishment, which is critical given the need to replace lost cells killed during infection. Lowered mucosal immunity decreases mucosal integrity, allowing increased translocation of microbes and ongoing inflammation. Existing infections could also reactivate in these circumstances. The ongoing replication of pathogens causes chronic production of pro-inflammatory molecules. All of these factors lead to chronic immune activation and exhaustion even under ART suppression. Therefore, studying immune cells, particularly those in the lymphoid tissues, can give us two important insights. First, how does HIV cause chronic immune activation at the Center for Immune Regulation and HIV Replication? And second, how do CD8 T cells interact with the infected CD4 cells in the tissue reservoir sites? So with this background, we will then move to the paper. The paper that will be the main focus of this journal club today deals with this topic of how the immune system responds to HIV infection and was published just a few weeks ago. Though this paper does not contain spatial techniques, it is at the forefront of single-cell analysis in the HIV field. In this paper, the authors want to identify the key immune responses that occur during the earliest stages of infection before and at peak viral load. This information has the potential to lead to prophylactic and therapeutic strategies for HIV. The samples in this paper were obtained from the FRESH cohort. And the FRESH cohort is a group of women in South Africa who participate in education programs and twice-weekly HIV testing. Some of the samples were collected before the standard of care in South Africa required immediate ART initiation. Instead, ART was begun if CD4 T cell counts fell below a particular threshold. As a result, these unique samples are able to show multiple months of acute infection. To obtain the massively parallel single-cell RNA sequencing that forms the backbone of this paper, the authors used the seqWell platform. To briefly describe the method, beads covered with barcoded oligos are loaded into wells with one bead per well. Cells are loaded into wells ideally with one cell per well, and the array is covered with a membrane with a 10 nanometer pore size to prevent diffusion between wells. Cells are lysed and mRNAs are captured by the oligos on the bead in that well. Beads are removed from the array with the captured mRNAs attached, and mRNAs are reverse transcribed to cDNA. The cDNA can then be amplified, prepared into libraries, and sequenced. With this technique, the authors sequenced transcriptomes from PBMCs collected from 4 FRESH participants. Analyzing multiple time points before and after infection, the samples show the expected increase in plasma viremia and the expected decrease in CD4 T cell counts over time during infection. In total, they captured over 59,000 cells after filtering for quality and averaging nearly 2,000 cells per participant per time point. To annotate clusters, they compared differentially expressed genes based on known lineage markers and previously published datasets. The data was similar to well-established PBMC subsets and distinguished subsets of cell types, such as antiviral, inflammatory, and non-classical monocytes. Using the combined data to assign cellular identity, they did not observe many participant-specific features, indicating that disease biology, shown on the right or on the left, rather than technical artifact, was the main driver of variation for determining cell type. They confirmed the frequencies of CD4 and CD8 T cells throughout infection with flow cytometry and monocytes with counts in whole blood. Next, they examined how different cellular phenotypes changed over time in acute HIV infection. Unlike previous single-cell RNA sequencing studies that used pseudo-temporal ordering to track cell fate decisions, they developed a framework to see how each cell changed phenotypically during infection by adapting weighted gene correlation network analysis. This allowed them to discover gene modules whose expression varied significantly over time with unbiased single-cell resolution. Because of the small sample size and the heterogeneity of disease responses, they analyzed each participant individually for cellular responses associated with plasma viremia, modules that group together over time, and possible molecular drivers and signals between cells. The gene modules showed common temporal patterns during infection. Starting with participant 1, they identified 6 modules spanning multiple cell types that all show their highest relative module score at peak viremia. Though these modules were in different cell types, they included overlapping genes stimulated by interferon. These expression patterns show cell type-specific genes that correlate with a shared overall function and core interferon-stimulated, or ISG, signature. They did not detect direct expression of interferon-1 genes, but they characterized the expression of an upstream regulator, IRF7, as a predictor of cells potentially responsible for interferon production. In participant 1, 6 of the 8 cell types showed higher expression of IRF7 at peak viremia compared to pre-infection and 1-year time points, emphasizing the key role of interferon in the early immune response. The other 3 participants each had plasmacytoid dendritic cell responses and ISG gene modules similar to participant 1 at or 1 week before peak viremia. Across individuals, common ISGs were shared in 2 or more participants, while some were specific to a single individual. To confirm the presence of interferons and downstream cytokines, they measured interferon gamma and IP10 at peak viremia. All 3 participants had higher interferon gamma and IP10 at peak viremia, and 3 showed elevated MIG. There was increased soluble CD14, a molecule associated with monocyte activation. All of these results indicate the common interferon response that occurs very early in infection as viremia peaks. They next looked for shared expression dynamics. With fuzzy C-means clustering for the median module scores at each time point across all cell types on an individual basis, they generated clusters of modules called metamodules. These metamodules represent gene programs across cell types with coordinated temporal dynamics. Metamodules were labeled sharp positive, sharp negative, gradual positive, and gradual negative to describe their patterns, with a few exceptions showing more complex patterns at the bottom. The sharp positive metamodule, or MMSP, contained the majority of the ISG modules. The only other metamodule spanning 5 or more cell types, metamodule sharp negative, was enriched for ribosomal protein coding genes, possibly indicating senescence of some cell types. To discover conserved modules across individuals, they bin time points by viral load. Most of these gene modules were consistent with the participant-specific approach, but this approach was unable to show any gene modules with consistent expression trends in 3 of the 4 participants, indicating the importance of participant-specific analysis to understand the variability. Looking more closely at the gradual positive metamodule, they noticed that it consisted of responses sustained throughout acute infection, but with different cell types in each participant. In participant 2, MMGP consisted of monocytes, B cells, plasmablasts, cytolytic T cells, and proliferating T cell gene modules spanning programs such as antigen presentation, IL-6 and IL-8 production, and granzyme B production. These temporally overlapping gene modules may represent cell subsets responding to common stimuli and/or to one another. Based on known relationships between genes within and across cell types, they use this information to generate a network model that describes potential axes of cell-cell signaling, including direct signaling such as via receptor and ligand, or indirect such as cytokines and chemokines. For a specific example, IL-6 and IL-8 from B cells and plasmablasts could attract monocytes that present antigen to CD4 T cells. This could upregulate IL-17 and BCL2, restricting cytotoxic T cell mediated killing of infected cells. The IL-6, IL-8, IL-17 signaling axis could thus be a target for future study in HIV treatment. So then they looked for acute infection responses that were present in multiple participants. Some gene modules were shared by 2 or more participants in certain cell types, while others did not share any genes across participants. They qualitatively compared gene module functional annotations within the metamodule gradual positive for each cell type across participants. The temporal dynamics and the specific genes involved varied, but there was significant enrichment for at least 15 of the same underlying pathways and functions in at least 2 participants. Thus, there may be common features in spite of individual heterogeneity in infection response. They hypothesized that there may be a shared set of immune drivers that coordinate gene responses to infection. To identify these drivers, they generated a list of predicted upstream drivers for each gene module in MMGP. From hits that were significant in at least 2 gene modules, they built a network of putative upstream signaling. Rescoring cell types against enriched genes for each driver showed kinetic variation in the onset, intensity, and length of immune responses across cell types. In all participants, CD4 T cell activity peaked from before peak viremia throughout acute infection. CTL and proliferating T cell programs occurred during hyperacute infection, and NK cell and monocyte activity persists throughout the first month of infection. The shared immune responses emphasized CD4 T cells throughout the first month of infection, as this was the only cell type expressing genes downstream of pro-inflammatory cytokines. Therefore, CD4s may be an important responding cell subset to cytokine production in early HIV infection. Gene modules with similar temporal response patterns within the same cell type, but with different pathway enrichments, imply orthogonal biological functionality. In one example, NK cytokine signaling gene module 3 and cytotoxic gene module 4 in participant number 3 show the same temporal pattern. To understand how these gene modules are linked, they looked across single cells for co-expression of these 2 modules. The correlation between module expression across single natural killer cells changes with time to decrease in later infection. K-means clustering separated cells based on this variable expression of gene module 3 and gene module 4, suggesting NK cells have a plasticity and can express both programs at peak viremia. Looking at metamodule sharp positive, they observed participant 3 exhibited temporally similar modules in monocytes that did not correlate variably over time. They were highly co-expressed only at HIV detection. Gene set analysis revealed monocyte gene module 1 was antiviral, while monocyte gene module 3 was inflammatory. So monocytes in participant 3 at the time of HIV detection were simultaneously expressing both the antiviral and the inflammatory gene programs. These polyfunctional monocytes were not identified in any of the other participants. Comparing genes in non-classical monocytes at peak response time points, they noted more participant-specific differences. Monocytes in all participants produced antiviral factors, but only in participant 2 and participant 3 did they note an enrichment of inflammatory responses. Because chronic inflammation has been associated with susceptibility to infection, they looked for markers of inflammation in participants before and after infection. They found variable inflammatory gene expression at the pre-infection time point and mixed changes during hyperacute infection, again highlighting differences between individuals in inflammatory state at a given time. Natural control of HIV has been associated with diverse cellular phenotypes in CTLs and dendritic cells, so they next looked for connections between disease progression and polyfunctional monocytes in participant 3. Both participant 3 and 4 maintained low levels of viremia, less than 1,000 viral copies per ml, by 2 3/4 years after infection without ART. These controllers could have enhanced systemic immune responses that enable long-term control of the infection. The CD8 T cells are known to contribute to this control of chronic HIV, so they analyzed them and noted increasing levels of perforin and granzyme during acute infection, indicating potential importance of CTLs in early infection control. Unsupervised and directed approaches did not show any further significant differences between groups. To determine whether the proliferation of these T cells was important to the long-term outcome, they looked specifically at proliferating T cells. However, proliferating T cells express similar levels of cytotoxic genes compared to the non-proliferating CTLs. They then looked at T cell receptor sequences to map clonal dynamics during infection. T cell receptor pull-down and enrichment showed a few expanded clones, but was inconclusive due to their small sample size, with only 982 total cells in which they captured the TCR. The controllers did display higher frequencies of proliferating cytotoxic cells within the first month of infection compared to pre-infection versus non-controllers, indicating that these proliferating cytotoxic cells could be important in control. Finally, they used unsupervised analysis to find differences between these proliferating T cell responses over time among participants. Clustering over all of these cells, they identified 4 subsets of cells with distinct gene programs: traditional CD8s, hyperproliferative CD8s, naive CD4s, and CD8 negative but TRDC and FGCR3A positive cells, which they determined to be natural killer cells. Based on the distribution of cells within each of the clusters, the NK cluster contained the highest proportion of proliferating cells at HIV detection and 1 week later, and most of these cells were from controllers. This suggests that a strong proliferative response in cytotoxic NK cells early during infection could have a role in long-term control of HIV. In conclusion, this paper used a novel single-cell RNA sequencing based framework and a unique longitudinal cohort to characterize conserved immune response dynamics, cell subsets, and gene programs during acute HIV infection. They discovered interrelated temporal gene module expression patterns in distinct cell types and predicted mechanisms by which multiple components of the immune system may respond in a coordinated manner to HIV infection. They were able to hypothesize when and how various cytokines, chemokines, and transcription factors could orchestrate immune responses during infection. Their work could provide a unique reference dataset for studying the earliest moments of HIV infection following detection and could suggest new roles for monocytes, NK cells, and CD4 T cells in acute infection of HIV. A key advantage of this single-cell approach in the study was the ability to identify cellular subsets present during hyperacute HIV infection in controllers compared to non-controllers. This includes polyfunctional monocytes in participant 3, and as I mentioned before, the subset of cytotoxic proliferating NK cells in both controllers. These proliferating NK cells could function with the cytotoxic T cells in early infection to mitigate the antigen load for the cytotoxic T cells, thereby reducing subsequent exhaustion. They suggest follow-up studies in non-human primate models to determine the importance of early immune responses for long-term viral control. Comparing these samples with the fresh cohort samples currently obtained from individuals receiving immediate ART compared to delayed ART could reveal the effect of early ART on immune dynamics in humans and add to the information from non-human primate models. While this study does not provide any spatial analysis and relies solely on peripheral blood samples, Like most studies in the HIV field to date, it demonstrates the wealth of information that can be obtained from longitudinal samples during the course of infection. The single-cell resolution is crucial to determine these key subsets of cells that respond in different ways to infection and lead to different infection outcomes. Additionally, the heterogeneity between individuals is a constant consideration. The small number of individuals here, especially with 2 controllers and 2 non-controllers, is clearly not sufficient to make any generalizable conclusions. However, this paper does provide hypotheses that can be tested in additional samples and mechanisms that can be examined in the non-human primate models and additional human samples. Given the potential usefulness of this study in spite of the caveats, using only blood samples, a small sample size, and simply looking at the transcriptome from these samples instead of multiplexing with other modalities such as protein surface markers, it is clear that spatial omics approaches in HIV would be a major source of new information and understanding. So while decades of research have taught us much about HIV, many questions remain unanswered. Some of the main biological questions are, what is the reservoir? Where is the reservoir? Is it in particular tissues or cell types? How is the reservoir maintained, either through long life or proliferation? Are cells preferentially located in specific areas of tissues? Do infected cells and tissues differ meaningfully from those in peripheral blood? And if so, what are these differences? What are the interactions and their effects between infected cells and those cells surrounding them? Does HIV infection alter the architecture and cellular composition of tissues in biologically meaningful ways from tissues in uninfected individuals? These questions all culminate in the overarching work towards a cure. How could the reservoir in tissues be targeted for a functional or complete cure, and would this strategy need to be distinct from that for peripheral blood? So the first few questions have been answered at least partially so far, but those in tissues are still extremely unclear. We need to address these gaps to move the field forward. Doing so will require innovative approaches to deal with the difficulties of HIV research. In particular, technical limitations in the biology of HIV infection present daunting challenges yet to be overcome. The largest challenge, as mentioned many times throughout this presentation, is the rarity of infected cells in ART-suppressed patients. In CD4 T cells in the peripheral blood, only 1 to 10 per thousand cells contains a provirus, and only 1 to 10% of these are replication competent, leading us again to about 1 per million CD4 T cells in blood with a replication competent provirus. Because of this, it is difficult to obtain enough cells from infected individuals to be able to make generalizable conclusions about the reservoir. The second challenge, particularly for spatial studies, is the availability of samples. For tissue samples, we've used small biopsy sections or tissues collected post-mortem. Autopsy samples bring the further complication of delay between death and collection, thus allowing RNA to degrade. Also, some individuals stop taking ART prior to death, leading to viral rebound, and this prevents the description of the ART-suppressed state. Furthermore, there are not many SNAP-frozen samples available relative to the many archived formalin-fixed and paraffin-embedded samples. As FFPE is incompatible with many spatial omics approaches and produces inferior RNA sequencing results, simply obtaining materials of sufficient quality for experiments can be a huge difficulty. To overcome these limitations, the field needs high-throughput techniques to rapidly and reliably detect proviruses in cells, even when the virus is latent, and ideally, they could be performed on FFPE samples. This may involve improving in situ hybridization techniques. However, the largest area of new research requires spatial context. If we grind up tissues to isolate single cells and correlate measures such as the frequency of HIV-infected CD4 T cells versus the immune effector CD8 T cells, we lose the opportunity to examine their interactions. Only if we look at these cells in their original spatial organization can we understand what is happening in the HIV reservoir and how to form solutions such as breaking immune sanctuaries. So the DBIT-seq method, developed by Dr. Fan's lab and described by Yang in the seminar last week, is an exciting possible solution for spatial transcriptomics and proteomics, including in FFPE samples. With resolution approaching the single cell level at 10 microns, we would be able to identify and spatially map cell types in tissues with the potential to simultaneously detect HIV RNA in actively infected cells. Our lab plans to collaborate with Dr. Fan to investigate the architecture and composition of tissues, focusing especially on the gut and the lymph nodes in HIV-infected individuals. Using RNA-scope and DNA-scope on adjacent tissue sections, we could better understand the spatial distribution of infected cells and the general immune landscape in these important reservoir tissues. Comparing the transcriptomes of immune cells in blood and in tissues could give a much more complete look into the immune system and the effect of HIV infection than has been obtained in any previous study. The rarity of infected cells, specifically the replication-competent proviruses, is still a key hurdle, but the high-throughput nature of recently developed single-cell methods and ongoing technological advancements are promising for the future of HIV research. With that, I would like to thank my lab for their support, especially my PI, Yachi Ho, and Dr. Fan for letting me spend time in his lab to learn about his new device and method, as well as inviting me to do this journal club. I want to thank you for listening to my presentation, and with that, I would be happy to take any questions you might have.
So thank you, Christine. It's an excellent talk. And so anyone has question, you can either unmute yourself and ask, or you can use the chat box. I think before people putting the question in the chat box, maybe I can ask one first. I noticed you talk about the CD8 cells in sort of controlling infected or latent cells, but I'm just curious how, for example, the antigen-presenting cells or dendritic cells, how they recognize the infected cells and send the signal to CD8 cells, or that completely bypass the sort of dendritic cell and antigen-presenting step, but just a direct communication between infected CD4 cells and cytotoxic T cells.
Yeah, so I guess the infected cell could directly present the antigen to the CD8 T cell.
No, I think my question is really how the CD8 cells, the cytotoxic T cells, recognize the infected cells, and the typical immune recognition mechanism is the antigen presentation by dendritic cells or macrophage. Even in the paper you discussed, it doesn't seem like people see any difference in the dendritic cells and how they play a role in HIV.
Yeah, I wouldn't say I know too much about that area at the moment.
So dendritic cells and macrophages present HIV to CD8 T cells from MHC1, and CD4 T cells can directly present infected HIV to CD8 T cells from MHC2. So they both present but separately.
Oh, okay.
Yeah, but they would only be able to present if they're actively infected, which is the issue, or they have a provirus that's able to produce the protein to be detected.
So any more questions from the audience? And it's a terrific immunology seminar, and I'll probably some kind of most audience today, they're either technology development experts or like a tissue biologist. And this is an excellent talk. I think I really learned a lot. Thanks, Kristen.
So Rong, this is Yachi. I have a question for you. Are you thinking about using spatial transcriptomics in COVID-19, or do you know of any research groups trying to do this?
Oh, this is a great question, but I think the policy right now at Yale and I think in entire Connecticut is we're not allowed to do autopsy. So tissue samples are not available, but now probably somewhere else, not here. But that's not urgent. So yeah, I'm interested, but that could be something we can work on later.
Because so once you can get those frozen samples, but once they're put in formaldehyde overnight, that you can take care of them in your lab safely. So it's great that you can work on fixed samples and not worrying about all these like droplets generated by 10x machine, for example. And say so right now all the bioarchive studies are published using say bronchial alveolar lavage, which is great, but it's still different because you don't see people are arguing that who is making IL-6. So everyone is claiming their favorite cell type is actually just macrophages and dendritic cells. But if you can just see them in the spatial context, I think that will solve a lot of these arguments and find the relevant therapeutic targets for either cytokine storm or viral replication.
Yeah, that's so true. I even try to argue we should do lung biopsy early on rather than just do post-mortem patient autopsy because at in that stage all the organs fail and what you are learning is not really how disease develop. But
I see. So the problem with lung biopsy is that once you do the lung biopsy, there's a hole. When there's a hole, then you're at the verge of respiratory failure. Once you create a hole, it's like you pop open a balloon. So that's why no clinicians will give you those samples because, well, this is already a terrible balloon. I'm not going to pop this open just for a study. So
that's right.
I suppose it's going to be very hard to get worldwide any samples at early stages of COVID-19. But yeah, so autopsy samples are likely by the end. But I agree with you that by the time of autopsy, there are all these like exudates and everything all messed up together. It may be hard to tease out which cell is making all those cytokines because it's just all messed up and necrosis and I agree. But, maybe we can have animal models.
Yeah,
they're monkey models.
That's right.
Yeah, monkey models. Well.
All right. So I wonder any more questions from audience, and then we have Kristen here. We also have Yachi here, and who are very interested in infectious disease. I believe nowadays everyone, if not HIV, and any questions you want to ask? Okay, if not, I think Yachi and Christine, thank you so much. And I recorded a video, and if you guys are okay with that, I can also post on our YouTube channel, and people who cannot join today can still have the chance to watch.
Thank you. Thank you,
Yeah,
Ron, for organizing this.
Yeah.
It's wonderful. Thank you.
Thank you very much. Thank you very much. So again, we have one more seminar next week for the series 1, but we already have a wonderful panel of speakers lined up for the series 2. Please go ahead and check out our Google Doc, and I think the entire series 2 is completely scheduled, I think, until I believe July 17. So many more exciting talks to come, and thank you for your participation and attending the seminars, and look forward to your continued support. So thank you, and take care.
Thank you.